Afternoon, everyone. My name is Armin Zakarian, and I'm the Vice Provost for Research at University of Michigan Dearborn. And I want to welcome you to our second of four UM-Dearborn, UM-Flint Collaborative Research Series event. I want to thank all the participants for taking time this afternoon to join us for the event, which is dedicated to engineering and healthcare. I especially want to thank the 8 faculty speakers who agreed to share their research with us today. Before we get started, a few more thanks should go out, and I want to express my gratitude to 2 chancellors, Chancellor Dada and Chancellor Grasso, for providing support for collaborative research grants in making this event series possible. Finally, I want to recognize and thank my collaborators, Office of Research and Economic Development at UM-Flint, especially Heather Dawson and Ken Sylvester, and also the Office of Research at UM-Dearborn, Wes Clark and Rebecca Rand, for organizing and hosting the events and managing the joint internal funding opportunity. I want to remind the attendees that besides today's event, there will be 2 additional sessions and networking opportunities for faculty. The next event is scheduled for March 11, same time, and focus will be on educational innovation, artificial intelligence, and computer science. And our last event is scheduled for March 25th, again, same time, and the focus is on social justice and community engagement. So everyone who is interested in participating in these sessions, please go ahead and register for these events. I want to mention that all our events are being recorded, and shortly after conclusion of each event, they will be available for UM-Dearborn and UM-Flint community to view. The UM-Dearborn and UM-Flint Collaborative Research Grants Program has been accepting applications since January 28. So anyone who is interested in submitting an application, they don't have to wait until the deadline, which is the April 25th, 5 p.m. You can start working your application and submitting it to the program using the InfoReady website. And I think Wes and Rebecca will share that link with you. The program is open to collaborative teams of researchers from both campuses. The requirement is that at least 1 PI from each campus should be involved in the application. Applications can address any research area that is of interest to the team. There are no topic limitations. So although we have identified like 4 different sessions, but those are just to facilitate the, kind of a networking. So we are not limited to some of these areas that we are discussing in these events. So our hope is that these events will provide the attendees opportunity to learn about the research of their colleagues across the campuses and find collaborators, to submit an application to the program. So the 8 lightning talks will be followed today by breakout sessions when all the attendees will be sent into breakout rooms, and we'll have the opportunity to discuss research ideas with colleagues from both campuses. Again, all the lightning talks will be recorded. The breakout room discussions are not recorded and will not have moderators. So it's a free flow discussion. Our sincere hope is that the conversations will continue after the event. So please exchange your information, and if you want to discuss further, schedule follow-up meetings to talk about possible research ideas. To support the post-event contacts and conversations, the Office of Research created a research profile directory, a place with the brief research profiles of faculty interested in finding collaborators for the joint grant program. I think that link will be available to you through the chat. And if you are not in this directory and want your name to be added to the directory, then you can email. I think the email is also will be placed in the chat for you for the contact information. Now we are now getting into our most exciting part of the session, the lightning talks. We had some really exciting and interesting talks last time we met. And we have 8 faculty who will presenting their research today. The first presenter is Professor Amanda Escobar. Amanda is an associate professor in the mechanical engineering department at UM-Dearborn, and her research is in bioengineering. So Amanda, you have the floor. I think you are muted, Amanda.
Okay, there. Should be all set now. Thank you very much, Armin. This is my information right here, and I am going to very, very briefly talk about injury prevention in sport. Hopefully everyone can see my screen. I just noticed. Let me move that. Okay, so I'm going to talk about 2 areas of research that I'm currently focusing on. The first is related to concussion and head injury, and the second one is related to knee injury prevention. Oh. So this first study that I want to briefly talk about, I'm going to pause that so it's not kind of distracting. So just, very, very brief background. Brain injury can occur in sports due to head acceleration events that happen when the body or when the head is either directly impacted or indirectly through the neck or the torso. You have like a whiplash type of injury. Both of these motions to the head cause high acceleration changes, both linear and rotational acceleration. And this can cause strain in the brain tissue, which then can lead to some type of injury that can cause either the tissue to fail or, can cause concussions and other head injuries that we're familiar with. So this particular study that I'm going to talk about that we're looked at, we specifically looked at the sport of lacrosse. So lacrosse is played by both males and females, but the sport differs for the 2. So for males, it's a collision sport. That means that they're allowed to purposefully make contact or hit each other. For girls and women, when they play lacrosse, that's a contact sport. I mean, they're going to make some contact with each other, but they're not allowed to purposefully do that. So because of that, the personal protective equipment differs. So boys and men wear helmets, with the hard shell that look like football helmets. They wear pads, gloves, things like that. Female athletes who play lacrosse only wear eye protection and goggles and mouth guards. But a lot of head injuries do occur in girls and women's lacrosse. So because of that, there's some newer soft headgear that's currently on the market. Now, this soft headgear does not have a typical hard shell. It's made out of foam. So what we were looking at was, we just kind of did an initial study looking at the efficacy of this. And we wanted to look at it at kind of this higher velocity, this 5 meters per second. It's a little bit higher. And we looked at, we impacted the head form in all of these different locations, 6 different locations. Actually, I just have 3 listed, both with no headgear and with the headgear. And we looked at different brain injury measurements that are used. So you can look at peak linear acceleration, something called HIC, which is head injury criteria, which is also related to linear motion. But then we also looked at angular acceleration, angular velocity, and something called BRIC, brain injury criteria, which is calculated using angular velocity because studies have shown that those correlate very highly to brain strain, which, like I mentioned, is what's going to cause injury. So just very briefly, I'll just kind of show you a little bit of our results. So you can see if you just look at this average number here, the black bars are no headgear, the white and the gray are with the headgear. So you can see, you know, for peak rotational acceleration, that actually decreased it quite a bit. Not as much with peak rotational velocity or brick. And then it did decrease our linear metrics quite a bit as well. So, you know, that was kind of interesting. This was a small pilot study just done with a couple of headgear. Other things that we're interested in, so lacrosse season typically can start in early March, which as we know can be very cold. So because these are headgear that are really made out of these foams, I'm also interested in looking at the effect of cold temperatures on these. I'm also interested in looking at the risk of injury to athletes that are not wearing the headgear. You know, is there some sort of increased risk of injury to them if they're playing with some athletes that are wearing them? And then kind of some other areas of interest that are related to head impact, but not sports. One of the things that I'm interested in looking at are head impacts in intimate partner violence, and also head injuries that are associated with falls in the elderly. So just kind of some future areas of research as well. So the other study that I wanted to briefly mention was looking at anterior cruciate ligament injury. So the ACL is a crossing ligament in your knee. It's located inside of your knee. It helps prevent something called anterior tibial translation. So that's like, it helps prevent your shin bone from moving forward. And, you know, it's an important structure. It's also a structure that is injured a lot in sports. And the ACL can be injured due to contact, right? Someone hits someone in the knee, but more often it's due to non-contact. So basically athletes land wrong and tear their ACL. Now, it's mainly been thought of as an acute injury. So you land wrong once and the ACL tears, but there's actually some evidence that in some cases, this might be an overuse injury. So maybe you're landing kind of wrong over and over and over, putting strain on that ACL until it finally fails. So if that's the case, one of the things that we're interested in is using wearable technology to potentially identify injurious loading cycles. So the wearable device that we're looking at is something called an IMU, or inertial measurement unit. It has accelerometers and gyroscopes in there. And so for the first part of the study, what we did is we looked at this cadaveric model, and we had, we used a cadaveric knee where we loaded it up to 100 times at kind of lower non-injurious loading cycles, and then higher loading cycles that are considered injurious. And by higher, I mean like 3 to 4, landing at like 3 to 4 times your body weight with an internal tibial torque at the knee as well. And so we're kind of looking to correlate, can we basically estimate some important things like ground reaction force, internal tibial torque, valgus moments, things like that we believe are what caused this injury. Can we sort of estimate that using an IMU? Okay, so that's kind of our question. So we're going to do this kind of in this cadaveric model. Then we're going to bring athletes into the lab. I have a motion capture lab where we're going to ask them to do you know, jump and land and run and things like that, and see if we can estimate some things with the wearable sensors. And then the last step will be a small validation study in the field as well. So that is just a very, very quick, I think I got my 5 minutes of some of the research that I've been doing. And I won't be in the breakout rooms, unfortunately, but I'd be happy to chat elsewhere. Thank you.
Thank you, Amanda. So we will be moving on to our next presenter, Professor Yasser Abul Qasim. Yasser is an assistant professor. Is an assistant professor at the University of Michigan-Flint, and his research focuses on cardiac mechanics, bio-inspired microtechnology, and microfluidics. That was a tough to pronounce for me. and also the innovative cardiac assistance devices. Yeah, sir, you have the floor.
Thank you, Armin. Good afternoon, everyone. Before I get started, I would like to thank the organizer committee and administration from both campuses for supporting to our research and starting this initiative. So let me start by a motivation slide here. So as you can see, one goal from my lab is to better understand the cardiac mechanics and how mechanical dysfunction of human heart can lead to different cardiomyopathy. This map illustrates for you data over 40 periods of or 40 years in the United States here for patients that they suffer from cardiac disease, and you can see how highly they lead to mortality. So the mortality rate is very high here. And if you look at the data carefully, you can see that we have about 600,000 that they die every year from cardiac diseases. And before they die, we spend about $400 billion every year to try to treat them. Most of these diseases are related to coronary arteries or mechanical dysfunction of the heart valves, which is they normally used to regulate the blood flow between the heart chambers, or due to the alterations in the conduction system or the electrical pathways in the heart, which it can lead to fibrillation or arrhythmia disease. So here I'm listing 4 projects that they are open for collaboration. These projects span out to different or 3 areas of research. One of them is a basic science or using computational modeling and mathematical tools to develop or to better understand the cardiac electromechanical coupling of the human heart. And the goal of this project is to establish or to build a simulation platform, what is normally known as digital twin. So we can move mathematical modeling and computational tools to a clinical setting, and then we can help the clinician to do better assessment or a better diagnosis. So the second project, which is cardiac optogenetics. So optogenetics is commonly known in the neuroscience community, where you can use light in order to alternate the electrical activity of the brain. You can use optogenetics to treat epilepsy, but we are adapting the same techniques into cardiac community, where if you look here, this is an isolated cardiomyocyte, it's just a single cell of a heart. And we can introduce opsins. This opsins is coming from bacteria. It's a specific protein, which here we use channel rhodopsin 2. This specific protein, when you shine light on it, it will stimulate the cardiomyocytes. So now your cardiomyocytes are open for dual stimulation, electrical stimulation as well as the light stimulation. So the expected outcome from this project is using light as a stimulus can be an innovative way for suppressing arrhythmia, for example, or you can alternate the mechanical contraction of the cardiomyocyte. Also, people trying to use this as a way as a regenerative medicine. For example, if your myocardium is not functional well, so we can do the myocardium, we can inject those cells with this specific protein, and then we can stimulate your myocardium with light as kind of mechanical pacemaker. So the third project here, or what you see in this image, is an X-ray imaging. Once we injected bubbles inside the tracheal tube of one of the insects, and we see how this insect, when they do body motion, they were able to move the blood flow with an efficient way to the distal locations until the oxygen will reach to every cell. So inspired by this system, we're trying to build a new micropump that we also would like to use it as an assistant device for treating people with this ejection fraction. So the fourth project is I'm trying to build on the expertise that we have for micro devices is looking into a micro stents. And we would like to focus our attention into the tissue stent design, because many of the design of these stents can cause a lot of inflammation in addition to clotting. So the design itself of how these stents are built It's very central in my research. And it's just a new project I started. I'm trying to do fluid structure interaction and then looking into what happened to the tissue in that regard. So I'm gonna give a brief focus on project number 1 and project number 3. So why do we need a multi-scale modeling when you develop computational tools? First is most of the interaction of these proteins at the molecular scale can give rise to a lot of biomarkers at the phenotype. So what we do as for a patient that they suffer from cardiac disease is we just go and do MRI scans, and we can see from the MRI scans that, okay, you do have a growth and remodeling for your tissue here, you do have some abnormalities in that place, and so on. We can also solve the blood flow inside the cavity using 4D MRI, and we can solve an inverse problem to give you the blood flow motion inside your cavity. And we can identify some of the biomarkers inside your cavity, but again, this is at the phenotype. But we don't know how these diseases are triggered from the molecular scale. So that's why if you are thinking to build a pipeline or a platform as a computational tool, you really need to know what's happening at the molecular scale here. What you see here is an interaction between two important proteins at the myofilament, which they are responsible for the contraction or mechanical contraction of the heart. These proteins are namely the tropomyosin, and this is it's like a helical coil around the backbone of the actin filament, and they are regulated by the calcium. And there is also some decorating proteins around the tropomyosin. So they work together in order to give rise to what you see at the phenotype or at the whole organ simulation. So if you can get the chance to introduce or to make use of the data that coming from the interaction of these proteins, and you try to propagate this data at the whole organ simulation, then you probably will be able to introduce some of the disease in your simulator and see what's happening at the phenotype. So basically, we have a lot of mathematical tools and simulators that can look at the whole organ simulation and also at the organelle or the cellular level. So again, using computational medicine, which is a new emerging area of research, people were able to do a lot of research in that space here. But at the subcellular and the molecular scale, we normally use molecular dynamics and coarse graining approaches to look at the interaction of these proteins. And unfortunately, we didn't have in vivo techniques to look at this interaction. So most of the work, they use the motility assays, or they can use in vitro experiment to look at how these proteins interacting to each other. So my goal is to look into how we can make use of the data at the subcellular scale, and we try to propagate it for at the whole organ at the tissue scale, and look at if we can propagate this disease at and look at it at the phenotype. So this is how I can see the platform to build the whole organ or the tissue scale. We started by MRI scans. We do segmentation and image processing to find where is the blood pool and the tissue segments. And then we do registering for the myocardium whenever we have a scar region. And the scar region is like a non-conductive region. So it's a substrate for arrhythmias. And then we build our finite element model. We do tagging for each region based on the NIH segmentation. And also we identify the fiber around your heart, and this fibers is responsible for the contraction of your whole organ. And then we do simulation at the whole organ using the finite element. But the question is this simulation is reliable? Is it accurate? So the answer is it is not, because well, I can do the same for a car or for your mug, for anything, right? Just a finite element is a useful tool for that. But it's the way that we do it right now, or people have done it over 20 years or more, they are not super accurate. So this is just inaccurate simulation here. You do you solve your bidomain equation, partial differential equation, to get the electrical activity in the heart. Once you base from one location, you calculate the activation time, take the activation time, and use a feed forward for your finite element to see how your heart is contracting. Well, that looks like a heart, but it's not functioning like a heart. You see, it's very softy, and also you get some wave propagation along the short axis here. So it looks like heart, but it's not beating like hard. So what is missing here? The missing information is how these proteins are interacting at the subcellular scale or at the molecular scale. So we did some work to identify what would be the energy landscape of how these proteins are interacting with each other. And we were able to provide a coarse-graining approach for the first time. There is the first time study that I spent for 6 years to come up with this energy landscape profile to be able to look at how these proteins are interacting and how the tropomyosin is moving over the active filament when it gets activated. And then we take this data and we did something called elastic network modeling. It's in a biomechanics. People that do biomechanics are very familiar with the elastic network, where instead of using the molecular description of each unit in your or each amino acid of your protein, we just approximate this as nodal points with finite mass and also with some of the springs. And then we solve for the eigenvalues. We get the eigenmodes of your tropomyosin, this protein, how it's moving. So here you can see a bending mode. Here you can see the torsional mode over there. We take this data and then we put it back in our finite element model. And then you can see now it looks like hard, but also beating like hard. So the whole idea of building this platform is to come up with a contraction model that is accurate enough to be giving you the synchronization motion of the contraction. So in the, I'm going to be in the breakout room to talk about the optogenetics in details, just for the sake of timing, but I would like to give you just a brief, again, introduction. So the idea is to take this channel rhodopsin, which is mainly located in bacteria, and then we transfer this bacteria or this channel rhodopsin, which only allow the ionic current, which is a potassium inward current for your ion channels. And like, once you get this perturbation of the additional ion currents, it's going to change your action potential. And then you can alternate the action potential of your electrical activity of the heart by just stimulation this using light intensity at a specific frequency. So it's a very neat idea. And again, it's adapted from the neuroscience community from Carl Lab at Stanford. So the third project that I want to really talk about is about the technology. So here, carpet beetles, the shaded area here is the tracheal tube. And we know that this beetle is having whole body motion, likewise a local contraction along the tracheal tube. And the question is why this insect is doing that. We find out that they do this specific motion with very accurate contraction or collapsing along the tube in order to convect the oxygen to downstream locations. And so it works like a perfect unidirectional pump, and it's also micro pump because you look at a few millimeters or to up to a few microns downstream here. It doesn't only happen at the abdominal tracheal location, but also near to the head and also the distal location to the bottom here. So it happened all over the place. And again, it's just to enhancing the convection mode of the oxygen to reach to the distal location for enough perfusion of the oxygen. So I find one German company is trying to do the same with collapsing sites of membranes. And then I looked into their actual design. Okay, yeah, so here we're trying to build a similar, using similar techniques, starting from the insect as your bio-inspired system. We look at the tracheal network and all the bifurcation daughter vessels here. We're trying to use the similar structure to build our network of microfluidics using elastic tubes. And then we focus our attention into one of the locations for us to use it as again, as a micro pump that hopefully will be implanted inside the chamber of the heart to enhance the ejection fraction. So their design is very complicated when it comes to valves, because mechanical valves are normally cause an issue. They go under fatigue because they are under dynamic loading, and then with time they will break out, and then you have to throw out this. But it's a portable module. You can just take this one, you can connect it to your microcontroller, and then you can use it for both gas and likewise for liquid. So this is already in the market. And if I just show you the design, so imagine that this is your tracheal tube, and you get two collapsing.
Yeah, sir.
Yeah.
Yes, sir. I think we are short on time, so if you can
Okay.
wrap it up, I think.
Yeah, I'm gonna do this right now. So we hope that we can get rid of those mechanical valves, and then we came up with our design here that just you have two contraction sites, and without any valves, if I can time how they contract and how they expand with certain time lag, I was able to create unidirectional flow. So the advantage here of this new design is just getting rid of your valves. So I'm going to be in a breaking room. If you have any question, I'll be happy to answer. And thank you for your attention.
Thank you. Thank you for that interesting presentation. I think we're going to move to our next presenter. Our next presenter is Professor Joe Lowe. Joe is an associate professor of bioengineering in the mechanical engineering department at University of Michigan Dearborn. His research interests are in biomedical devices and also biophotonics. Joe, you have the floor.
Okay, thank you, Armin. I'm gonna pop up my slide there. I was just thinking, asking a professor to present their work is like asking a 6-year-old to show their favorite toy. Everything is very interesting. So I'm hopefully not going to overwhelm you too much. I want to highlight several of my research projects, but also paint a picture of the theme of my lab. All of my bioengineering research involves trying to improve or discover new mechanisms in diabetes and diabetic therapeutics. So the first topic that I want to talk about is related to biophotonics. I'm interested in looking for a way to non-invasively detect tissue collagen in tissues related to diabetic wound healing or tumors and scarring. The way that we go about this is that the collagens in the extracellular matrix will fluoresce a particular wavelengths and also signature of light if they're excited by the UV light. And this non-invasive biophotonic information can be best quantified not just by the wavelengths itself, but also by the temporal information. And we construct this homemade fluorescence lifetime imaging system in order to detect the different compositions of collagens in tissues like normal skin or wound skin or tumors, etc. So apologize for the sort of grotesque picture that you are seeing right now. This is a foot ulcer from a diabetic donor. The areas highlighted was the numbers written in there are their fluorescent lifetimes. What we found is that the ulcer area had longer lifetimes, and they are associated with the more immature type 3 collagen than the regular type 1 collagen that is in the stronger tissue. So these ulcers, if you're not healing them carefully, even though on the surface might be closed, this one is obviously not closed, it might look close, but it'll open again because the underlying collagen is not strong enough. So hopefully a non-contact microscopy scan of the underlying collagen could help tell the physician whether or not to advise the patient to lay off that area of their tissue. The interesting problem is that we collect lots and lots of data, and if we just represent them in spatial terms, that's great, but we actually have too much data to just put into one number for a particular tissue. So I have to do different ways to represent the data. And one inspiration that we got was to draw from the way that the the circuit people represent their information in terms of phasor plots to tell you about the temporal information in their circuits. And this is sort of like the circuit analogy for these wound ulcers. Different phasor plot can be plotted for wound tissue and normal tissue, healed tissue, or like sort of the tissue that look healed but is not ready yet. So that's the one of the interesting question that I need help on, is that we're starting to get more information than we can process, and there are probably better ways in addition to what I'm showing here of representing and trying to analyze the data from biophotonic information. The next couple of topics that I want to highlight are my device work. This particular device is a droplet array that senses the different amount of antibodies related to diabetes. And these are using porous hydrogels in order to detect the influx of autoantibodies. And what we're interested in this particular project is to learn how to better control the size and shape of these particles so that we can make even smaller droplets and encapsulate them with cells, for example. The next project area is related to this idea that I started looking at diabetes epidemiology. This is a map of the health outcomes of children in Michigan, and on the right here, well, center here is the same map showing child poverty in Michigan, and you can see that it's almost a very unpleasant correlation between poverty and health outcome, and some of this is related to diseases like diabetes. So poverty related to obesity, sedentariness of children's activity, so on and so forth. So our project that I'm highlighting next is to sort of look at dietary and or environmental factors, that is free fatty acids in the bloodstream and hypoxia from the tissue, and see if there is some sort of link to explain something like this kind of phenomena that we're seeing using microfluidic devices. And my hope is that once we have found some sort of collaboration, I could extend these parameters to whatever other epidemiological factors that I can consider in addition to fatty diets and lack of exercise. The last area that I want to talk about is the area of microfluidic pumping, and I tried to draw inspiration from previous scientists, like all of us do. So I want to talk about the other Nikola Tesla inventions, because Tesla is known for his work in electricity and motors, but he's also a very productive inventor in terms of fluid mechanics. So this is one of the pumps or rotors that he designed in the early 1900s. It's a rotor that he used to generate hydroelectric power. That's why you saw that picture at the Niagara Falls that I just put up. What I found was that I like to leverage the fact that this Tesla rotor pumps out very smooth flows. So we're thinking to miniaturize it in the micro domain to get a miniaturized pump, and we were able to do that. Then I delved a little bit deeper and looked at, well, can we improve Tesla's invention by adding topology on the surface of these rotor disc in his rotor in a way to improve the coupling of the fluid to the surface. And this is sort of simulation, and we have additional experimental data now to show that yes, there is something that you can do to change the boundary effects of the liquid and the solid. So here, what I'm hoping to get is some sort of collaborative idea or help on modeling this interaction between fluids and surfaces, and both experimentally and in terms of modeling in order to move this work forward. So at very end, I just like to say that besides all of the stuff that I've shown you, I also try to participate a lot in outreach programs. So that might be another thing that we could find collaboration is outreach events that you're doing and that I'm doing that we could combine together. I know Amanda is trying to create one of these new programs right now, so I'm going to talk to her about it. But yeah, thanks for listening to me, and then you could please contact me during the breakout sessions to talk about any of these topics further.
Thank you, Joe. Thank you very much. Now we'll move to our next presenter. Our next presenter is Jennifer Blackwood. Dr. Blackwood is an associate professor of physical therapy, and she's also therapy department director at University of physical therapy department director at the University of Michigan-Flint. Her research interests include relationships between cognitive function and falls in older adults, population and age-related changes in balance function in older adults. Professor Blackwood, you have the floor.
Thank you, and thank you for the opportunity to do a presentation today. Just to make sure folks can hear me, I was doing this with my students, and that you're seeing the right screen. Are you seeing my slides? Thumbs up if you are.
Yes,
Thank you. Okay, so this is my lightning talk. I'm honored to give the talk today, and of course, I can't say any of this stuff without all the collaborators, students, and faculty that I've worked with over the years, and my mentors. So thankful for them. I'm going to tell you a little bit about one of the areas that I like to look into, and this relationship between cognitive impairment and physical mobility and falls, and specifically because it stems from a lot of the research that I've done in geriatrics that has identified that specific areas of cognitive deficits contribute to deficits in not only physical mobility, but changes in gait and increased risk for falling, as well as an increased risk for frailty and falling. And a lot of the research that I've done previously has really just looked at this relationship in older adults, and we find these significant associations, and it's become this thing that's more common in physical therapist practice and really in medicine, is to try to address this concern of falls as it relates not just to strength deficits and maybe balance deficits, but also how cognition plays a role in that. And more specifically, my role has really been to start investigating this and looking at this in relation to our older cancer survivors. So why is that an issue? Because not only are these individuals older, they some of them have this sort of genetic precursor for some other concerns relative to having a diagnosis of cancer, but also their pre-morbid status might put them at a risk for higher balance impairment and a greater risk of falling. But when we throw in this idea of not just having a cancer diagnosis, but more importantly, the treatment for their diagnosis, we run into things, as you can see on the right-hand side of the screen, such as chemo fog, chemo brain, essentially changes in cognitive function as a result of most generally the treatments, if you will, to sustain life as a result of cancer diagnosis, but contributes to a higher risk for falling. And this is an area that really hasn't been studied much previously. So some of the work that we've done in the past is on the top left-hand side of the screen. And it's really looking either at large data sets and looking at relationships first with that to identify how specific components of cognition are associated with falls or balance and mobility impairment in older cancer survivors. So we used the health and retirement study for that and found significant associations between executive function, which is kind of our main area that we tend to look into, but also some areas within recall and orientation in these relationships. And then because in physical therapist practice and really in healthcare, we have to make sure that we're using tools that are valid in certain populations. And in physical therapy, we often have these measures that therapists are using to assess fall risk and assess balance. but we didn't know whether or not they were actually reliable and valid. And so a number of papers have come out producing that material in older cancer survivors, and specifically for cognitive assessments in this population. And then lastly, right before COVID, we had a nice study that was going on looking at physical performance measures and how they differed in individuals with the history of cancer diagnosis, in particular those individuals with impaired executive function, because a lot of the literature really points back to these deficits in executive function where you're able to do more than one thing at one time, if you will, in the very generic definition of that, and its relationship with maintaining balance and fall risk, and as well as gait deficits. So, COVID-19 hits and things change a little bit. And so since that time, we're starting to get reignited and restarted in some of these areas. But I have a PhD student right now that's working on a systematic review of the non-pharmacologic interventions to address cognitive function in older adults, in particular, the components such as physical exercise or computerized cognitive training. I've done some work with the computerized cognitive training in the past, and we found some unique relationships between a computerized version of like Lumosity, if you will, and physical performance in just general older adults. But I want to see if that happens in our older cancer survivor. And then the other thing, sort of the big wish, if you will, is to really look at how some of these evidence-based fall prevention programs and balance programs may contribute to better performance and decreased risk of falls in our cancer survivors. So my contact information is on the top right-hand side, and that is a picture of lightning over the Lake Michigan, which I would not want to be on that boat, but if you want to be on the boat with me with one of these studies, please meet me in the breakout room. Thank you.
Thank you very much, Dr. Blackwood. Our next presenter is Gargi Ghosh. Gargi is Associate Professor of Bioengineering in the Department of Mechanical Engineering at University of Michigan Dearborn. She directs the work of the Soft Biomaterials Lab, where with her research assistants, she is working on the development and characterization of polymer networks with desired structure, property, and functionality for a series of applications in biomedical field. Gargi, you can go ahead with your presentation. I think you are muted, maybe. Oh, I can hear you now. Yeah.
Okay. So yeah, as Armand just said, I am interested in biomaterials. I mostly work with soft materials like the hydrogels, and we look into different applications of this material. So overarching, if you look into the overall theme of my project that's going on, so we are trying to develop 3D models of tumor and trying to look at their angiogenesis properties or switch that's happening, or also their metastasis to different organs. Also, we are into tissue engineering. So we are looking more into controlling the behavior of the stem cells, more from the perspective of their paracrine signaling rather than differentiation, which is mostly people interested in. We have worked a lot on this, developing different hydrogen microparticles for diagnostic purposes, mostly some cancers and all. And we have worked, like me and Joe collaborated in some of those projects. Not been concentrating on those right now that much, but if you're interested, obviously you can ask me anything about that. So I'll be talking mostly about the other two projects, like the tumor and the mesenchymal stem cell stuff right now. So as far as the tumor project is concerned, so our hypothesis is in most of the solid tumors, as the tumor progresses, the external matrices undergo remodeling, resulting in the stiffening. And this stiffening of the matrices actually helps in the shift of these the cancer cells to more of a proangiogenic switch. They become more proangiogenic because they need those blood vessels to survive and grow in size. And our hypothesis, if that's happening, maybe inhibiting the cells to sense the stiffness probably will be a way to prevent this vessel formation, blood vessel formation, reduce their vessel density. So with that, what we did is in our lab, we used collagen as our scaffolding materials. We changed the stiffness without changing the collagen concentration, because that's a problem because we are increasing we do not want to increase the concentration of the collagen, density of the collagen. So we actually what ended up doing is pre-glycolating them with collagen, and depending upon the concentration of ribose we were using, we could tune the stiffness of the matrices. And then we embedded the cancer cells within these collagen gels. And we did observe that when the cells were inside the stiffer gels, their secretion pattern changed. So a lot, when we compared the secretion of, we actually collected the conditioned media, tested tons of this different angiogenic and pro and anti-angiogenic molecules. And then what we found, most of these pro-angiogenic molecules, the secretion was much higher when we are looking into the matrices, which are much stiffer compared to what they are in a compliant gel. So the one that we are looking at, the compliant, is that the stiffness is more close to the stiffness of the breast tissue. On the other hand, the stiffer is what we generally see in the breast cancer extracellular matrix tissue stiffness. So we compared that, and that's what we saw, that actually there's upregulation. So right now we are looking at whether if we can inhibit the signal, the cells to sense this changing matrices, will that change the pro-angiogenic activity of the cancer cells? And obviously going forward, if that happened, we would like to test them in animal models, or we like to test them with different types of other cancer cells. That's where I would like to have collaboration, someone who can help us actually doing the animal study for us to see whether we are saying in vitro is actually true in real, in animal body or not. So that's what we are as far as the 3D tumor model is concerned. The other project that we are looking are mostly in tissue engineering, because as we know, there's a huge gap between the demand of damaged tissues and organs and actually that's available. And so these are the ones numbers that have thrown in and actually taken from the information that's only valid for US, not even global. And there's a huge number of people, around 25% of the people who are in the wait list, they actually die without even getting up the list to get an organ transplanted. And that's what's prompting this tissue engineering field to develop these tissues in vitro in the lab so that they can be used. We have them off the shelf. So one of these cells that people primarily use is the stem cells, because obviously the field started because these cells can become any type of cells that we want them to be. They can start from the stem cells and we can make cardiac and any lung tissue, whatever skin, whatever cells we want to. But what we found, people have found out, when these cells are injected directly into the damaged site, 90% of the cells are lost. Only 10% are at the present at the site, which is not enough to regenerate the tissue. And the second thing that's from those studies, people found out it's not only the ability of these stem cells to differentiate into other type of tissues, it's that this ability of the stem cells to release some molecules which actually attract our own body cells to the damaged site and help them to heal that damaged tissue. So actually, these cells are by releasing these molecules, they're helping our body to heal themselves. So what in our lab we are interested is how can we change, again, because we are more into cell material interactions, changing the matrix properties, we can control the release of these molecules that the stem cells are releasing. And because if we can do so, we can control the rate of tissue growth. So we have been looking at both the tissue stiffness. So this is one of the studies where we actually just used polyethylene glycol diacrylate and gelma, which is gelatin methacrylate. We combine them together, make different type of gels, we change their stiffness, and we found the stem cells, they actually prefer an optimal stiffness for this maximal release of some of these growth factors which are necessary for this tissue regeneration. We also are looking into the ECM's composition. So for this, we grew the cells under different culture conditions, decellularize them, characterize the matrices, and then throw in the stem cells. And then what we find that depending upon this ECM composition, the secretory profile of these stem cells are changing. So they are not same if we are growing them on well plates or we are growing them, or even between the ECMs, the secretory patterns different. Not all the data out here, but that's what we saw. We also looked at the bioactivity. So because it doesn't matter if we are seeing a lot of this different change in secretory activity, if it does not translate into actually something happening with the tissue regeneration. So we looked into the blood vessel formation in vitro. So it's a tube formation angiogenesis assays. Again, we found that what's reflecting in the conditioned media, that when we are using the conditioned media coming from the cells which are seated on specific ECMs, the tube formation is much more higher. We also use that. We actually, because we are interested in the wound healing of diabetic patients, so what we actually tried to mimic in the lab is we created some hyperglycemic fibroblasts, the skin cells like the fibroblasts, the connective tissue generative cells that's helping us with the generation of collagen and elastin. So we created these hyperglycemic cells, but we found like their functions are impaired. Then we actually incubated them in the presence of these different conditioned media, and we kind of saw the recovery of their functions. So we are kind of now planning to take it to more into the skin explant study to see actually what happens if we put them in the explants that we are trying to get from some of like human skin explants where we'll be putting them. But we also want to look into some other aspects, because I know this is important when this vessel formation activity is important in case of myocardial infarction, where we have tons of cells that are dead and we need to recover them. But there the problem is we have this anisotropy as well as the stiffness. So we are going to look more into them, and any further collaboration in that field, like helping us out with that field, really, because we do not know much, because we want to see how that will help in the, if we put these stem cells and anisotropic matrices, how the secretion will help in the maturation of cardiomyocytes and all. So any help there will be really helpful. So with that, I will be there in the breakout room. Any questions, anything that you want to ask, you can ask me there, or we can chat about this. So that's it.
Thank you, Gargi. Thank you. We are moving on to our next presenter, Dr. Marilyn Filter. Dr. Filter is an associate professor of nursing in School of Nursing at University of Michigan-Flint. Her research focuses on women's health using photo voice methodology and quantitative and qualitative methods. Dr. Filter, you can go ahead.
Hello, thank you for allowing me to do this presentation. And I'm following a little bit different format because I'm a nurse, and nurses are practitioners. And so oftentimes what we find comes from a completely different place than a lot of other researchers. So what this is the story of two advanced practice nurses, an engineer and a tech advisor, who've set out to change the world for women's health. I'm trying to get this to go forward. Okay. This is our team, myself, Lynn Benke, Lynn Van Neustadt, and Mary Auger, and we'll talk more about them as we go. We're going to take our trip. We were all nurses, and I've been in women's health. I'm a certified nurse midwife for many decades. And there were things that bothered me the whole time I was in there. And one of the things that bothered me were speculum exams. And if you talk to any woman who's had a speculum exam, she will probably not get up and cheer and say that was the favorite thing she's ever had done. And when we did the research, we found that many women avoided getting screening for cervical cancer because they did not want this procedure of having a speculum. And so our burning clinical issue is there a way that we could replace the speculum for vaginal exams? And you need to know, speculums have been around for over 200 years. No one's ever changed it. Problem being, though, in women's health, we're underrepresented in research, and there is also very little reimbursement for procedures that are directly related to women's health. So that drove our team member formation. So I found Lynn Benke, who is an advanced practice nurse. She happened to be on the U of M Rhodes Scholar bus and was talking to someone else on the bus. And we found our partner, Lynn Van Neustadt, who was then engineering at Dearborn. And so when we say our team formation can be formal or informal, it's just amazing how we have formed our team along the way and found team members. Mary Auger, who is our senior tech advisor on our campus, was really instrumental in connecting us to many, many resources. And we also want to give a shout out to our student teams, because for a while we had a GRSA working with us and helping us through. Currently, we're working with BME 599 students on the Ann Arbor campus who just have been doing a phenomenal job of coming up with other ideas to help us format where we were going. So how did we do it? Now, mind you, 2 years ago, I had an idea. I was a newbie, didn't have a clue how to go forward. So I did take it was suggested I take the fast-paced course from FFMI in Ann Arbor, which I did, which I have to say provides just such good support for you to give you some structure as to how to move forward with all of this. So I learned how to do customer discovery. And for many people, customer discovery, they don't realize how important it is. So I went around to people, both providers and consumers, and I said, what do you like about having a speculum exam? What do you not like about having a speculum exam? What would you change if you could change something about this particular exam? Now, those who have had a speculum exam kept saying, I don't want to be put in a vulnerable position. I don't want to be put on my back with my legs spread out and someone's face in my perineal area. So both the consumers said they didn't like that. Providers don't particularly care to be sitting in that particular close proximity to the orifice of the woman's body. So it was uncomfortable for most. I also then went and looked at the literature and said, what happens to women who've been violated when they go into the emergency room and they need specimens taken? We found they were put in the same vulnerable position. We found that women who had female circumcision had very small orifices and speculums did not fit into their vaginal openings. And lastly, we found in the LGBTQ+, we found that older men who are males who have sex with males, their rectal tissues became very taut and not pliable. And so they did not tolerate rectal exams. So then we made our, we worked with Tech Transfer, and Tech Transfer walked us through every step of the way. What do you want to do? How do you want to do it? They walked us through all the NDAs, you know, with all of the companies we worked with, because we had to, one, figure out what was currently being used. And of course, standard of care for cervical cancer screening is a speculum with a brush. So we then looked at, had anyone else changed from a speculum to another device? We did our research in that area, and we found that there were some companies that had like endoscope pieces, but they did not collect cells. And this is very different from having tissue taken. So then where did we go? Now, you have to write grants for funding. We were very naive in this whole area. And so we have to give thanks to the Office of Research, you know, for pointing us in directions of where can we get money. We looked for small internal and external grants to help us through the way. working on some federal grants, and then we found some philanthropic foundation grants along the way. We were successful in funding in a couple of them, not successful in a couple of others. But again, how many grant agencies are interested, number one, in women's health, and two, replacing the speculum that's been around for 200 years and it works, so why change it? Another challenge we had was prototypes, because we're two nurses. So it's very interesting how these two nurses started out in hardware stores and big box stores trying to find things, pieces, parts that put together. Because of this very sensitive part of the body, it's very interesting that the commercial simulators weren't really lifelike. And we ended up finding ourselves on websites that nobody really wants to look at. But, we have found simulation tools that work for us. And so that's how we started with prototypes. We caution everyone when we got our first grant and we're going to work with a commercial prototype company, we were advised to stay away from that expensive prototype company in a particular area because they were going to be way beyond, your grant funding. And we went with another prototype company. That prototype company did not provide our expectations of prototypes. So now we've written another grant and we're moving forward. But you need to be aware when you're looking for all of these things. The Office of Research on our Flint campus has been wonderful, but we have gone through MISHAR on the Ann Arbor campus. MISHAR has their own branch of CSCAR that they work with. We have worked with patent attorneys, which is another whole world, is working with patent attorneys. So we do hold a patent for the FemScope. Okay, there we go. So what did we do? We went to the statisticians. We said to them, what is it we need to prove to get this to market? Statisticians laughed at us, said, you'll be lucky if you get 25 women who volunteer to allow you to have this procedure done on them. We said, okay, that's done, because every woman we've spoken with would volunteer to have this new procedure done because there was such a difference. We talked to IRB because this is a vulnerable population. This is a women's health issue. We've had multiple conversations with them moving forward. We are in the midst of getting our prototypes for both simulation and live or human studies. For us, animal studies didn't make sense because we would have had to change the actual size of the device if we were going to utilize it. And none of us wanted to take the animal training needed to do animal studies. So we decided that we would go directly from our simulation to human studies, and we're planning on doing that starting in the fall. In the area of commercialization, when it comes to doing this type of research, you have to decide, do you want to do a startup or do you want to do a licensing? If you're young and you're adventuresome, I'd say do the startup. Those of us that are on this team tend to be mature people, and we felt that licensing was the better way to go. And so when you talk about that and you talk about licensing and you talk about how you're going to work through patent and licensing, let me tell you, the people in Ann Arbor Tech Transfer MISHAR are fantastic with walking you through that. And so we talked about speculums. Okay, this is an actual picture of me holding the FemScope. For those of you who really don't know a lot about women's health, the speculum is an instrument inserted into a woman's vagina that stretches the vagina to about 6 centimeters. this femscope that we have developed, which is really an endoscope with some additional parts that we patented. And if you look at the end, you will see this end piece. We had to think about friction within the vagina. We had to think about cell collection because it's a particular type of cell that's retrieved at the os of the cervix. And so the last thing we found is as we move forward with all of this, because we were dealing with women and we were dealing with women's vaginas, a lot of people were very uncomfortable having this discussion with us. And because they were uncomfortable, we've had to desensitize many people to all of this particular type of research that we're doing. We do have other women's health research that we want to work on, but because the reimbursement is low, we have to come up with a device because reimbursement for a speculum exam is $50. So whatever we make has to be probably a one-time use. It has to cost less than $50. And what you also can't see is there is a camera and a light source on this endoscope. So when it goes into the vagina, we have a clear visual field of what we're looking at, as well as we see it on a tablet or a smartphone. So if anyone has any questions, I'll be in the break room. Like I said, I come at this from a very different way of looking at it because I'm a practitioner. And so I look forward to any suggestions that anyone has. Thank you.
Thank you, Marilyn. We want to move, again, we are running really behind. So if you can stick to 5 minutes, that would be greatly appreciated. I think we have two presenters left. Our next presenter is Natalie Sampson. Natalie is an associate professor of public health at the University of Michigan, Dearborn. Natalie's research focuses on community-based research, environmental health, inequity, disparity, and land use and health. Natalie, please, you have the floor.
Thanks, Armin. Thanks. And I'll try to be faster than lightning so that folks can actually get to chat for a minute before we wrap up today. And just thanks to everyone to hear what's going on our little old regional campuses is really heartening. It's amazing, impactful work. And Marilyn, make them uncomfortable. Desensitize us all. I love it. So as Armin mentioned, my work is related to environmental health, environmental justice. I threw up here on the screen a map of Southeast Dearborn, Southwest Detroit, Ecorse, River Rouge, for those who aren't from the area, and just really grounding you all in kind of what drives my research, which is, these 40 plus polluting facilities co-located next to one, about to be two international border crossings, and a lot of industry that goes with that and economic opportunity that goes with that. But with that, we know there are, we're in non-attainment for sulfur dioxide, and there's fugitive dust exposures, and there's PFAS in parts of the river perhaps. And so really just trying to figure out what data we need to drive policy. And for me, that looks like participatory research, action research, community-based participatory research, different names for it, but really working with fence line communities to build capacity around their data collection, or, whether that be me doing that with them or creating the tools and resources. So that could look like a health impact assessment designed to inform, the multi-million-dollar projects that are land use projects, infrastructure projects, that could be qualitative interviews, focus groups, trying to understand the scale and scope of some of these environmental issues. And so I'm happy to be with all of these engineers today. I do work a lot with toxicologists, engineers, landscape architects to try to wrap our heads around some of these issues. And I'll just say, really, again, the values that drive my work, if anyone could guess what these numbers are, just to be a little extra depressing on a Friday afternoon. So if you look to the Intergovernmental Panel on Climate Change or the Global Carbon Budget, these are the numbers staring us down right now, of trying to keep our temperature rise from less than 2.7 degrees Fahrenheit 11-ish years is what some scientists say before we are really getting into an area that we can't turn back from. And so those are the numbers that keep me up at night. Those are the numbers that remind me that there's not time for 10-year studies and getting papers out in 20 years from now. But if we're working on these issues, the most vulnerable, the most at risk, the most disinvested, excluded communities are going to be hit hard. And so I am reminded by a lot of environmental justice advocates that, you know, to really create that change, the research is following, you know, community mobilization, community organizing, advocacy that's happening for decades. And so up here on the top right, you'll see a group of youth that I am lucky to know. You'll also see Michigan's Attorney General Dana Nessel in the middle and some of our steering committee members for a project called Environmental Health Research to Action. So this project is with Dr. Carmel Price, who's a sociologist on our campus in Dearborn, and many, many other community partners who have been doing this work for a long time. We are focused on building capacity with these high school students. Each summer we spend time in intensive training. They're paid to participate. Many of them are from fence line communities to figure out how this all does go together. So everything from how do we already monitor water, air quality, soil, contaminants, heavy metals, how do we do all of that? What devices can we use? What is environmental health? What is environmental justice? All the way to taking that data that they collect and doing a policy advocacy training. So mapping out who actually can use this data and needs this information. Also storytelling training. So how do they tell their stories? So really matching the qualitative and the quantitative and the action and the research, if you will, to get it out into the world. And so it's been a really powerful experience. And so really just trying to think about some of these folks as future scholars who are off to be, you know, giving these lightning talks on their own and doing impactful research in the next 10 or 20 years. Whoops. So this is just skimming the surface here, but I wanted to kind of go back to those values I mentioned. And so for me, because of sort of the urgency and just the hustle that is academia, it is really important to me to try to get this work out in plain language, to try to get this work out with partners and disseminate it in ways that it can be used. So I just wanted to give examples from some different projects that I've been part of. And I can't begin to list all of the collaborators and the funders who've supported this work, but just showing how the priority for me has always been, you know, getting out, if it's an infographic, if it's a report, if it's multimedia, with the papers, of course, that follow, and advancing the scholarship and trying to bring that data to bear. So you'll see here on top, there's a link to several reports that came out of some green stormwater infrastructure work that I worked on with some folks in Northwest Detroit. There is a link to a YouTube video that talks about the ERA project some more, and you can see we also have a manuscript that talks about that process. A toolkit that's looking at some policy analysis that is local environmental justice work happening in California. And then finally, I mentioned that international border crossing. So part of the work that the community has been doing in Southwest Detroit for over a decade has been to really navigate what that means to live next to that. And so we've brought some data to bear to have conversations about how do we change the infrastructure in a way that can promote health. So whether that be building vegetative buffers, whether that be rethinking truck routes, right? And so some of that has entailed this citizen community science, but I've been lucky to follow the lead of a lot of amazing activists. So that is a whirlwind tour of my research, but I know we don't have a lot of time, so I'm going to stop there. Happy to talk to anyone from Dearborn, Flint, and anywhere across the state who's interested in building this research capacity with community partners, and specifically my passion right now with youth who can really bring some energy to this. So thank you, thank you, thank you.
Thank you, Natalie. And we are now into our last presentation. Our last presenter is Professor Nathaniel Miller. He is an associate professor of psychology at the College of Arts and Sciences at the University of Michigan, Flint. Professor Miller.
Okay, well, thank you for that introduction, and thank you everybody that was able to make it out today. It's been really great to hear the work that everybody's been doing. And so I was just gonna, I should go relatively quick today, but I just wanted to talk a little bit about a project that I've been working on with some collaborators, Linda Zhu and Charlotte Tang at the University of Michigan, Flint, in part because we're currently looking for other collaborators to help us validate validate and test a mobile phone application that we've been using to track Parkinson's disease symptoms. But I also decided to talk a little bit about this project just because it shows the dimensionality of my research program, and as we're kind of out there looking for potential research collaborators, some of the skills and abilities that I have that might lend to some of the research work that's going on across these campuses. So as I already said, we've basically been, I guess just to provide a little bit of background about myself. So my background, I'm a cognitive neuroscientist. And so basically what I'm interested in is looking not only at changes in behavior that are associated with different types of damage to the brain, whether it's through trauma or disease or a number of different things, but also what the neural mechanisms are that underlie it. And so it was kind of nice to hear a little bit about optogenetics today. But as part of that, the model that I've been using to look at that work is Parkinson's disease. And the reason for this is multifold, but one reason is there's an area of the brain that's damaged in Parkinson's disease called the basal ganglia that's responsible for a range of different behaviors. And we can actually see this if we look at the symptoms of Parkinson's disease, where there are the motor symptoms that we typically associate, such as tremor, problems with walking, problems with balance, but there are also a range of other cognitive symptoms that can occur. So problems with, like Jen Blackwood was talking about, cognition, so the ability to think, to plan ahead for events, to maintain attention. And then there's also a range of psychological issues, especially in Parkinson's disease, an increased likelihood for both depression and anxiety. And so this has provided a nice model in order to be able to test the role of the basal ganglia in these different processes. But personally, one of the reasons that I've also been interested in Parkinson's disease is I have a close family member that was diagnosed, and also several family friends, and kind of watching the experiences that they go through dealing with this range of different symptoms that can come up. Some of the big problems they face are having limited time with their healthcare providers. In fact, in some cases, for the people that are prescribing their medications, they may only see them 2 times a year. And then also, given the kind of wide range of different symptoms that come up, they have to work with multiple clinical providers, such as physical therapists, occupational therapists, speech therapists, to work on helping to mitigate some of their symptoms. And so basically there's this problem of requiring multidisciplinary care teams that there's not much crosstalk between them. And so kind of looking at these different issues and these different problems, it led us to start working on a project where basically what we're trying to do is to take an instrument that most people have of a smartphone and to really kind of leverage the tools that are already embedded within that cell phone. And so one of the things that I'm interested in is tracking some of the motor symptoms of the disease. And as has already been talked about, there are sensors within there that allow us to easily track that those symptoms, but then also leveraging some of the connectivity abilities that are in cell phones to be able to take regular surveys from patients. And so, you know, from a clinical perspective, the goal of this app is that it would allow the multidisciplinary care teams, caretakers, individuals with Parkinson's disease to hopefully improve communication and also allow for more regular testing of symptoms, for better medication management of those symptoms and whatnot. But then also from the research perspective, I myself, one of the things that I'm interested in is looking at exercise interventions for individuals with Parkinson's disease. And so this serves as a nice research tool in order to be able to collect that data remotely, so that we don't have to trouble people with coming into the lab regularly and all the hassles that are associated with that. So and at least for the Track My PD project, currently we've been working on some algorithms to measure things like balance, walking, slowed movement, and also implementing some survey measures in there so we can track other symptoms such as depression, anxiety, and some other psychosocial issues that can come up. And so, you know, we're looking for collaborators that can both help us to both kind of develop and validate the tool that we've been using, but then also the plan plan in the future, looking for collaborators that might be interested in doing some intervention work and using this as a tool to regularly collect data from their from participants in the study, whether that's Parkinson's disease or also some of the measures that we're including here could extend to a range of different diseases. And so for this project, it's something that I'm interested in finding some collaborators for, but then also kind of moving forward, working with some other individuals across the different campuses who might be interested in looking at some of the possible community-based interventions for individuals, and also tracking that. So sorry, it was kind of rushing because I want to give everybody enough time to be in the breakout rooms, but that's kind of a quick overview of the research that I've been working on, and I look forward to some of the discussions that I might be having with potential collaborators.
Thank you, Nathaniel.